Hello, this is Tommy. Welcome back to ChIP-Seq. In today's video, we are going to reproduce the final panels of figure 1 in a genomics paper. So make sure you watch the other videos in this playlist so you understand the context. And I will have all the R Markdown files and also the website link in the show notes of this video. Make sure you read that and follow along. So in today's video, we are going to reproduce this figure 1i and 1j here, okay? So if you remember, the authors performed ChIP-Seq for those three transcription factors, YAP, TAS, and TAT4. And in the figure 1i, they're trying to show, okay, the YAP, TAS, and TAT4, they kind of overlapping with each other, the binding sites, right? So we need to read the figure description to understand what it is and then understand how to make the figure. Okay, let's go to the figure description. So it says figure 1i, here is the heat map representing the overlapping binding sites located on promoters and the enhancers here. And the peaks are ranked from the strongest to weakest signal in the TAS signal. So in a window of 1 kb centered on the summit of the TAS peak. And this HCK4 monomethylation is an enhancer marker. HCK4 trimethylation is a promoter marker. They also show that actually those two data sets from a previously published data sets. In this case, we are not going to plot the raw signals here, but we are going to use the peak files that we downloaded from the last video and to determine what are the promoters and what are the enhancers for those binding sites, okay? And from the description of the methods, it says, okay, they're generally using a customized customer R script, which considers a 2 kb window centered on the peak summits and normalize the reads density with a resolution of 50 base pair, okay? So essentially figure 1i is just a heat map, okay? So if it's a heat map, what are the rows and columns? So a heat map is just a representation of a data matrix. And then you map the values, the data values to color, right? So according to the description here, each row here is one peak. And those peaks are overlapping peaks for those three transcription factors. And then each column here is one bin, right? As according to here, is with a resolution of 50 base pair, and then you have 2 kb window like here. So essentially you have, for example, 1 kb upstream and 1 kb downstream of the summit of the peak. And so here you, with 50 base pair per bin, so you have like 20 bins on the left and 20 bins on the right. So total you have 40 columns. Okay, now that we understand what is this heat map, and then we need to understand how to actually get that data matrix, okay? So you can actually use BAT tools and just given the overlap peaks with a BAT file, and then use the BAT tools can calculate the number of reads per 50 base pair being flanking this summit, right? You can use that, then you can plot that. But usually, especially in like now it's like 2025, this paper was published over 10 years ago, and now we have actually many other R packages or Python packages to do similar things. For example, deep tools in Python, you can create figures like that. But I always like R and try to get the data and into a matrix form and then use complex heat map to plot the heat map. Okay, let's go to R Studio and let's do that. Okay, so let's first just load the libraries that we need to use. So our track layer is an R package, bioconductor package to read in the BAT files. So remember, we call the peaks using max3 for those three transcription factors. So now we can import from the R track layer and read in those three peak files. So we can do that. And as you can see, those are just like peaks. For example, like you have almost 10,000 TAS1 peaks, and you have a couple of columns here. The most important is the chromosome start, the chromosome name and start and end, indicating the locations where TAS bind. Now we can find the overlapping peaks for those three factors. So now we have YAP over TAT, for example, YAP over TAT, use the subset by overlaps function from genomic ranges, I think, genomic ranges package. And you can subset. Now you find the YAP and the TAT's common peaks. And then you can use that to further subset by the TAT4 peaks. Now you get the overlapping peaks for those three factors. Now take a look how many. So now you end up with only 6,000 something, okay? Now, since we actually focus on YAP1, I will use the summit of YAP1 as the anchor point. Of course, you can change it to TADs. I mean, the paper is used the TADs summit, but it doesn't matter. You can use either one. So the summit, this file is also generated by Mac3. So from the previous video, I showed you how to generate those files on the command line. So make sure you watch that. So this is the file, the bed file for the summit. Summit is the highest point, signal point for that peak. So if you do that, look at, so it's just like one single base pair because it's the highest point of the signal. Now we use subset it to only the common peaks of these three factors we just calculated. Also, we need to read in the history modification peaks that we actually downloaded from Geo, only the bad files, and we actually lift over from HG19 to HG38. Make sure you watch the previous video. And those are just bad files. We use those bad files to determine what are the promoters and what are the enhancers. Okay, so we can define enhancers. So this, and we use invert equals to true. So H3K4 monomethylation, but not H3K4 trimethylation. So it's not in the promoter region. So it's the enhancer. And for promoter, is the other way. So you only have H3K4 trimethylation, but not H3K4 monomethylation, for example. So now you have the enhancers, like 22,000. And you have promoters. That's like 13,000 or 14,000. So now we can actually annotate the peaks, right? So we can subset by overlaps. This is the common peaks among those three. See how many of them are overlapping with the enhancers. And then we know those are the YAP enhancer. And we see how many of them overlap with the promoters. Okay. So you see we have actually only 274 promoter, and the majority of them are actually enhancers, like 3,488. Okay. Okay, let's look at the enhancer. Take a look. So it's just YAP peak name here. And remember, we need to subset the YAP1 peak, the summit as well. This is the total peaks for YAP1. Then this actually is the common, actually three factors common binding sites, and then annotate with enhancers. Take a look at here. This YAP1 overlap TADs and overlap TAD4. Okay, now we just use this to subset the summit of the enhancer and the promoter. And the anchor will be these two together, like the promoter and the enhancers, right? So essentially, those are the rows, all the rows here. Those are the anchors. And now we also need to import the bigwig files. Bigwig file is a pile-up file indicating the raw signal for each transcript factor. And I already showed you how to actually generate them in the previous video. Now we can import them. Also use the import function, read them in. The bigwig files are a little bit bigger, like a couple of 100 megabytes. So it takes some time to read in. And then once you import that, it's just a genomic ranges object as well. So let's take a look after it's okay. Now it's done. So if you take a look, it's just a genomic ranges object. For example, It tells you the locations and then also the score, right? And then you'll notice that actually we use the 50 base pair bin when we create the big wig files. You see here, this is the 50 base pair width, and this is the signal, the number of reads, and normalized by the total library size, and it's just a score, right? Now we can quantify the signal in bin, in this 50 base pair bin, flanking the summit of the YAP1 center, the summit. So use enriched heat map, and this is an extension of the complex heat map. So you use this function called the normalize to matrix and give the raw signal, which is the big wig file, the anchor, and the value column will be this column. And then we need to extend 1,000 base pair upstream and 1,000 base pair downstream, right? And then calculate the mean, and with width will be 50 base pair. So now you will end up with 40 columns, okay? So let's do that for the YAP1, TAD, and also TAD4. Okay, it takes some time. So once you actually create this matrix, so essentially this function is calculating for each 50 base pair bin how many reads, like normalize big wig signals in those bins, right? And you can think about what's the dimension of those three metrics, right? How many rows, how many columns? The number of rows will be how many actual anchors, right? How many actually rows do you have? This is 3,762. And the columns will be 40, right? So we can actually double check. Okay, the dimension for the matrix 1, matrix 2, matrix, they are always the same actual dimension. And if you look at the first 5 rows and all the columns here, then you'll see this. So now each row here will be 1 peak or 1 summit, one anchor, and flanking by 2 kb, 1 kb upstream and 1 kb downstream, right? And each column here will be just one bin, like you will see U1 to U20. That's upstream 20 bins, like because you have 1 kb divided by 50, that's equals 20. And you also have D1 to D20, that's actually downstream, bin 1 to bin 20, right? Okay, so now we need to map the values to color, then make a heat map. So let's look at the data actually range for those 3 metrics in order for you to map the values to color. So this is the quantile function. Okay, 10% is like 1.1. So above the 50%, the 90% would be like percentile would be 13.55. But you have a really big value here. Like if you use the maximum value is like 330. So let's do this. Okay, take a look here. So looks like majority of the value will be roughly around the 20 here. So that's why I actually used the circularize column RAM2 function to map white to red from value 0 to value 20. Okay, and anything that's above 20 will be just red. Okay? So potentially, like you can use this trick to remove the outlier skewness. So you can do that. Okay, now we also need to have this partition promoter versus enhancers, right? So we just use this wrap function. This how many of them a promoters is 274, and here is 3,488. So if you just do this, and this is just a character vector with like names, enhancers, or promoters. And now we can change that to factor and make sure the promoter comes first, because in the final figure, like promoters come first and the enhancers come next. So we can change the factors. And now we can make a heat map just for this partition here. So use the heat map function from complex heat map. And this is the color. Okay, and this, so if we do that, just create a heat map. The partition heat map, if you do this, it will show you this partition heat map. Okay, and now let's work on the main body of the heat map. So we can use enriched heat map function and give the matrix that we calculated. And then the color will be the color function that we mapped here, like this one, color function. And then there's some actually column title and the name. So let's do that. So the first heat map is that partition heat map, and the rest will be the matrix, okay? And it will be, okay, now we can draw that list of heat map and split by the partition. The main heat map will be the second one. Okay, let's do that. And this is what we get, okay? So we split by the partition, like this one, the factors. Like, so that's why it's split by the promoter and the enhancers. And then we use this main heat map will be this YAP1 heat map, okay? Okay, so that's exactly how you make a heat map from ChIP-Seq data. Now let's go to figure 1J, right? Okay, what is figure 1J here? Is this one. And let's look at the description of that figure. So if you look at here, it says this by model distribution of HK4 tram model methylation around the summit of this YAP TAZ and TAF4 peaks, okay? So it's actually just a line plot, right? And you notice that when we create figure 1I, okay, here, the line plot here is actually what we want, okay? The line plot here is just the average of all the peaks per column or per bin here, okay, here. So if you do that, okay, if you go here, so just the column mean of that matrix, mat1, and then the column mean of the second matrix for TAT, and then for TAT4, right? So if you look at that, it just give you for every bin here, right? So every bin here is the average across all the rows or all the peaks. And we can just plot the mean, for example, and this is actually what we want, right? This is the line plot. So we can actually put the three lines together as a data frame here. So like we can do bind rows, and it will be like that. And I can do mutate factor, so add a new column here as a factor, and then will be YAP, TAT, and TAT4. And if you do that, okay. So it will be like that, okay? So now we can make it into a long format, meaning use the pivot longer function from tidyr, and then all of those columns will be pivot into long format. If take a look how it looks like. Okay, so this should be in the dplyr. So it will be like that. So the factor, the names, and the value, okay? Now we can actually, now with that, we can make so this is the same thing as here. I'm just building the commands step by step. So now use ggplot to plot. So the the aesthetics will be, X will be the name, X-axis will be the name, and Y-axis will be the value, and then use the geometry line, and then the color equals to factor, and the group equals to factor. And let's see how it looks like. Okay, I need to dplyr as well. So, oh, something is wrong here, right? Why? It doesn't look. It's because if you look carefully, D1 now it's in the first, and D10 is the second, and ideally it should be actually up U1 to U20 and D1 to D20, right? So we need to change the factor level, okay? Change the x-axis order by changing the factor levels. So now just this command, I mutate that name into a factor, and the levels will be U1 to 20 and D1 to D20, okay? Now let's do that. If you do this, okay, dplyr, and let's plot the line plot. And this is what we want, right? Now I just do a little bit cleanup of those access labels and then the colors. And if we do that, Okay, this is what you get for a publication-ready figure. It's a line plot for those 3 transcription factors, and it shows you they all actually bind to the similar place. And this is the summit of the YAP1. Okay, and I think I will skip figure 1K because we have already done that in the previous figure. Okay, so that's actually about it for all the steps that are needed to reproduce this figure 1. And there are a couple of key takeaways here. Okay, learn the pre-processing steps for a particular NGS data type, in this case ChIP-Seq. And mostly it involves Unix commands, because we started with FASTQ files, we do quality control FastQC, align it to the genome, use about I2, and then Corpix use Mac3, and then make big files using deep tools. And they're all actually done in Unix commands. So learn how to use Unix commands. And then once we do the pre-processing, now we can dive into R or Python, and then analyze the figures, then try what type of the plot it is and what data needed, and what are the x-axis and what are the y-axis, and we can use R or Python to recreate those figures. Okay, and you see actually most of the time I was spending how to wrangle the data into a data frame or into a data matrix that is needed to plot, right, using ggplot2 or complex heatmap, right? And also you need to know probably which R or Python conductor packages to use. Always ask that ChatGPT. So now it's just much easier than the old days. Okay. And also once everything is in a data frame, usually for all this line plot, box plot, scatter plot, you can just use ggplot2 to make any figure you want. Okay. Okay, that's about it today, and don't forget to join my free newsletter here, and also subscribe to my ChIP-Seq YouTube channel too. And that's it, and happy learning. I will see you next time.